Hello everyone, welcome to my YouTube channel Microbiology Simplified. I am Dr. Amit Kumar Singh, MD Microbiology, working as professor. So this is the third video of Parasitology lecture series, and in this video I am going to discuss the other morphological forms of protozoan parasites. So without wasting any time, let's start with the video. So what are the protozoan parasites which have got morphological forms other than cyst and trophozoite? So these are hemoflagellates and sporozoa. The morphological forms seen in hemoflagellates are called as mastigotes, and the morphological forms seen in case of sporozoa, apart from trophozoite, are schizonts, merozoites, gametocytes, and oocysts. So, if you remember that hemoflagellates, basically there are two commonly pathogenic hemoflagellates are known, which are trypanosoma and leishmania. Okay, so in them they don't have any cyst or trophozoite. They have got typical characteristic morphological forms called as mastigotes. Okay, mastigotes means having flagellum. Okay, so they have flagellums. So there are four types of mastigotes. First one is amastigotes. Second one is promastigotes. Third one is epimastigotes. The last one is trypomastigotes. Okay, and these trypomastigotes have again got two different names. I will tell you later why it is. One is metacyclic and one is simply called as trypomastigote. Okay, so there are four different morphological forms seen in hemoflagellates: amastigote, promastigote, epimastigote, and trypomastigote. And in which flagellate, hemoflagellate, what morphological form will be seen depend upon the species. Okay, and the morphological forms seen in sporozoa are schizonts, merozoites, gametocytes, and the oocyst, which contains sporozoites. So they are also known as sporulated oocyst. Okay, so let's start with the first image. So if in this image, if you see, there is small rounded to ovoid structure can be seen. Okay, so these rounded structures are present inside a cell. This cell is a macrophage, and this rounded structure, if you see, you can see this is the nucleus and this is kinetoplast. So if I'll draw a diagram of this, so it will look something like this: a nucleus and kinetoplast, and then there is one more structure present in this which cannot be seen in this smear, is called as exoneem. This one is kinetoplast and this one is the nucleus. Shape I told you, it is rounded to ovoid in shape, and the diameter is approximately 2 to 4 micron. Okay, so these are the structure which is seen in this first form. So what this form is? This form is basically, if you see, there is no external flagella, nothing is outside attached to the cell. That means there is no external flagella. So this is actually a amastigote. This is an amastigote, which means no external flagella. Amastigote without having external flagella, but they have got an internal flagella which is called as exoneme, and this internal flagella originates from a structure called as kinetoplast. This kinetoplast is further made up of two parts. One is blepharoplast, another part is basal body. Okay, and this is the nucleus. The nucleus is situated posteriorly and this is the anterior. So in this case, the nucleus is situated posterior to the kinetoplast, or you can say kinetoplast is situated anterior to the nucleus, but there is no external flagella. Okay, so this is the amastigote form. Now coming to the next form, if you see, the shape is now not rounded, it is lanceolate shape. Also, one more point I want to add into that. This shape was, or this structure was initially called as LD bodies by the scientists who have discovered them, Leishman, Donovan. Leishman, Donovan bodies. Okay. Okay. So the second structure, second image is of, looks like lanceolate shape, and if you see, if I draw the diagram, it will look like something like this. Let me change the color. So in this, what is the difference between amastigote and promastigote? This structure, first is the shape, that is lanceolate in shape. Second, if you see, there is an external flagella. See, there is an flagella. There is an external flagella. Second thing, again this kinetoplast is anterior to nucleus. Okay, and this time the flagella is coming out from the anterior end. Okay, and there are two more structure of hemoflagellates in which you can see undulating membrane. There is no undulating membrane in case of promastigote. Okay, so among trypanosoma and leishmania, this amastigote form will be seen in leishmania and trypanosoma cruzi. Leishmania and trypanosoma cruzi. However, this promastigote is only seen in leishmania. Okay, so it is seen in leishmania species, but it is seen in trypanosoma cruzi but this is only seen in Leishmania species, not in Trypanosoma, any species of Trypanosoma. Okay. So now move on to the next structure which we can see in hemoflagellates. The first one is epimastigote and second is metacyclic trypomastigote. I have not shown any images for this because these two morphological forms are not present in human host. These are present in the insect vector. These are present in insect vector. Okay, and these two forms are not seen in Leishmania. These two forms are seen in Trypanosoma species. Okay, these two forms are seen in Trypanosoma species, and the species in which it will be seen is Trypanosoma brucei. Trypanosoma brucei. Okay. However, I'll draw the diagram of this epimastigote, how it will look like. This epimastigote will look like this. Again, elongated structure. Okay. And you can see the nucleus here anteriorly. But in this case, compared to this promastigote, the kinetoplast is little nearer to the nucleus, or we can say that it is little posterior compared to the kinetoplast of promastigote. And also the flagellum. Also the flagellum will not come directly from the anterior end, however it will run alongside of the body as a short undulating membrane and then come out from the anterior end. So in this you can see a short undulating membrane. If you remember, last time I told you about short undulating membrane in Trichomonas vaginalis. So you can remember that both Trichomonas vaginalis and Trypanosoma starts with T. So both have got short undulating membrane, which is basically an extension of cytoplasmic membrane and helps in movement. So this flagella initially runs along the body and form a short undulating membrane and then comes out anteriorly from the anterior end. And in this case, the kinetoplast is little closer to the nucleus, that is juxta nuclear. Okay. So these forms cannot be seen in peripheral blood smear. It can be seen only in the insect vector. Now coming to the next one, that is metacyclic trypomastigote. This metacyclic trypomastigote and trypomastigotes are morphologically identical. Only difference is the host. So how this metacyclic trypomastigote will look like? If you see this diagram, I'll just make another diagram for this to help you understand in a better way. So in this case, nucleus is not that posteriorly. Nucleus is almost centrally situated, and in this time the kinetoplast is not anterior to the nucleus, however it is posterior to the nucleus. Kinetoplast is posterior. See this, kinetoplast is here and nucleus is here. Nucleus is situated centrally and kinetoplast is situated posterior to the nucleus, and this is the anterior end from where the flagella is coming out. Flagella will always come out from the anterior end, and from that end you can determine that which is the posterior end. So if this is anterior end, this is the posterior end, and in this case the nucleus is situated posterior to the kinetoplast is situated posterior to the nucleus. Okay, and uh, flagella the flagella runs along the entire length of the body as a short undulating membrane before coming out from the anterior end. So in this case there is a long undulating membrane. Okay, and kinetoplast is kinetoplast is posterior and nucleus is centrally placed. Okay, so these are the four morphological forms which can be seen in hemoflagellates: amastigote, promastigote, epimastigote and trypomastigote. This epimastigote and trypomastigotes are only seen in trypanosoma and not in leishmania. So remember that in leishmania we will see only amastigote and promastigote, whereas in trypanosoma brucei, we will see epimastigote, metacyclic trypomastigote and trypomastigote, whereas in trypanosoma cruzi, we will see additionally an amastigote form, which is in case of trypanosoma cruzi is known as leishmanian form, and in case of leishmania, it is known as leishmania donovan bodies. Okay, so out of these, the intracellular structure is amastigote. Intracellular morphological form is amastigote, whereas rest of the form are present in the blood or in the tissue. So basically, hemoflagellate cannot survive outside blood and tissue. Okay, so because of that reason, or because they don't have cyst, they cannot survive outside the human body in an external environment. Okay, so now move on to the morphological forms which I have told you, which will be seen in case of sporozoas. The first one, if you remember, is the schizont. So this is the structure of schizont. Okay, this schizont is present inside the RBC, and it is almost spherical in shape, and it contains numerous small structures which are called as merozoites. So in a schizont which is produced during erythrocytic cycle or the erythrocytic schizont. Schizonts are of two types: erythrocytic. If you know the life cycle of malaria, you must remember that schizont is produced two times in the life cycle of malaria. One is during the erythrocytic cycle and one is during the exoerythrocytic cycle, which occurs inside the liver cells. So one is erythrocytic stage and one is hepatic stage, or erythrocytic schizont or hepatic schizont. The difference between hepatic schizont and erythrocytic schizont is that it occurs inside the hepatocyte and this occurs inside the RBC, and the number of merozoites, particularly in case of hepatic, it is in thousands, 4,000, 5,000 merozoites. However, in case of livers, erythrocytic schizont, the number ranges from 8 to 32 depending upon the species of the plasmodium, and it also contains a pigment called as hemozoin pigment. Okay, so schizont contains merozoite and hemozoin, and it is of two type: erythrocytic schizont and hepatic schizont. Difference between them is the first the location and second is the number of merozoites. Okay, then there are two more structure which can be seen only in case of this sporozoa is the gametocytes. So if you see, there are two different type of gametocyte. One is some like crescent shape and one is rounded. So basically the gametocytes in case of plasmodium are usually round in shape. They are usually round in shape, except in case of plasmodium falciparum, in which the gametocyte is crescent in shape. Okay, crescent in shape, or also called as banana shape. Crescent shape or banana shape. If you remember, what is the another structure which I have told you previously, which is crescent in shape, is the trophozoite of Toxoplasma gondii, which is also known as tachyzoites. Okay, and the gametocytes of plasmodium, females are larger than males, and females are called as macrogametocyte because of this, and males are called as micro. As they are large, so they are called as macrogametocytes, and male are called microgametocytes. So these two structures are formed inside the human body, and the formation of this is called as, from the merozoite, is called as gametogony. I'll make a separate video on the life cycle of malaria and all the diagnostic modalities which can be done, and the disease separately, because it is very difficult to teach the malaria along with other all parasites, because then the lecture will be too much lengthy, and then you may lose interest. So better to remember those points which are actually important for you to identify the organism first of all. Okay. Then there are two more structure which we can be seen but not in the peripheral blood smear, are microgametes and macrogametes. These microgamete and macrogametes are formed in case of plasmodium in insect vector. Okay, whereas in case of coccidia parasite, it is formed inside the human intestinal epithelial cells in case of coccidian parasites. Okay, so just remember right now that in case of coccidian parasite, the microgametes or macrogametes, that is sexual reproduction, occurs inside the human inside the intestinal epithelial cell. So in that case also we cannot see them in the stool specimen, and also in case of malaria we cannot see because these are formed inside the insect vector. So microgamete and macrogamete cannot be visualized in peripheral blood smear, but they are actually formed from the macrogametocytes and microgametocytes respectively. So now coming to the next morphological form seen in sporozoans. So these structure tissue cyst, and tissue cysts, and then all these four uh, images belongs to oocyst. So this tissue cyst and oocyst, this stage is not present in plasmodium. However, oocyst is seen in case of plasmodium in insect, okay, but not in human. So oocyst is present in plasmodium, which contains the sporozoite like the oocyst or this other coccidian parasites, but these oocysts are formed inside insect vector from the micro and macro gametes. Okay, and what is the insect vector? I'll make a separate lecture for that, but still I'm asking. The insect vector in case of plasmodium is female Anopheles mosquito. So just see this tissue cyst first. If you see this tissue cyst, first of all, it is round in shape. Second thing which we can notice, that this has got separate so many small structure. These small structures are basically rapidly multiplying brady, sorry, my mistake. It is not rapidly multiplying, it is slowly multiplying bradyzoids. Okay. The name itself, Brady, indicates it is slowly multiplying. So even if you forget about what is which one is rapidly and which one is slowly multiplying, just remember tachycardia means increase in heart rate and bradycardia means decrease in heart rate. So Brady means decrease, so slowly multiplying. So even if you are forgetting it, you will be able to recall that okay, you are doing some mistake, and then you can rectify it as I have done right now. Okay. And this tissue cyst is basically present in tissues of intermediate host in case of toxoplasma and sarcocystis. This tissue cyst is not present in this opportunistic coccidian parasite causing diarrhea. These are present only in the opportunistic coccidian parasite like toxoplasma and sarcocystis, and this tissue cyst are present in intermediate host. And in case of toxoplasma, The intermediate host is cat and other feline. And the commonest site of this tissue cyst in case of toxoplasma is brain followed by skeletal muscle and then any other organ. Whereas in case of sarcocystis, the intermediate host is cattle and pig, and this in them the commonest site is muscle. Okay, so nothing much to remember in this. They are round in shape, contain slowly multiplying stage called as bradyzoids. However, in case of toxoplasma, these bradyzoids are basically trophozoites. So again, easy to remember that in case of toxoplasma, these bradyzoids are trophozoite. T for toxoplasma, T for trophozoite. However, in case of sarcocystis, these bradyzoids are merozoites. Okay. So this is about the tissue cyst. Now coming to the oocyst. Oocyst basically contain the sporozoites, also known as sporulated oocyst. So all the opportunistic oxygen parasite causing diarrhea contains oocyst, including the toxoplasma and sarcocystis. They too have oocyst stage. Okay. So if you see the diagram or the image, you see this is in case of cyclospora. The name itself suggests cyclospora, that is rounded spore. The spores are rounded in shape. In case of cryptosporidium, the spores are present in the crypts, so that's the name is cryptosporidium. It is rounded to ovoid. That is, it is rounded to ovoid. And in this case, it is elongated or ovoid. That is cystoisospora. The cyst is elongated and ovoid. And there are certain more differences between them. So let's see the differences in a tabular form. So what are the differences between the oocyst of various coccygean parasites? So first see the shape. So the shape, the only little different shape is of cystoisospora, which is elongated and ovoid, whereas almost similar looking like oval or rounded in case of cryptosporidium, toxoplasma and cycloisospora. So remember this, elongated ovoid cystoisospora. I'll change the color of the pen. Cystoisospora has got elongated or ovoid. This will help you to identify the images given in the question, or even if it is mentioned that the cyst seen in the stool is elongated or ovoid, you will be able to identify that which is the cyst belong to which species. And another feature which is important to remember is the size. The biggest cyst is of cystoisospora. So elongated and biggest cyst around 25 micron in diameter or length is of cystoisospora, whereas the smallest one is cryptosporidium, 5 micron, followed by cyclospora and toxoplasma. So smallest is crypto, biggest is cysto. Smallest is crypto, biggest is cysto. Okay. And then the number of sporocysts and sporozoites. So what is a sporocyst? A sporocyst actually sporoblast which has got a cyst wall. When the sporoblast is covered with a cyst wall, it becomes a sporocyst, and this sporocyst contains sporozoites. And in case of toxoplasma and cystoisospora, the number of sporozoites and sporocysts are 2 and 8 respectively. Sorry, 8 and 2 respectively. Sporozoites are 8 in number and sporocysts are 2 in number. That means each sporocyst has got 4 sporozoites in them, in these two. However, in case of cryptosporidium, the number of sporocysts is 2, but the number of sporozoites are 4. That means in this case, each sporocyst has got only two sporozoites. So remember this, you have to remember this. And in cycloisospora, there is one sporocyst and four sporozoite as like toxoplasma cystis. So in case of crypto and cyclo, four sporozoites, but difference between cyclo and cryptosporidium is the number of cysts, and number of cysts is more in case of cryptosporidium, that is 2. And in case of toxoplasma and cystoisospora, the number of sporozoite and sporocysts are similar. Okay, so I believe if you remember this small table, you will be able to identify on the basis of image itself that this oocyst belongs to which species. Okay, and also these oocysts, that the oocyst of this cystoisospora, cryptosporidium and cycloisospora, because these oocysts are present in the stool or feces of humans. These are present in, these three are present in feces of human. However, this oocyst is present in feces of cat, which is the definitive host in this case. Okay, and human is the only host in these three. Okay, so in feces of human we can find only the oocyst of cystoisospora, cryptosporidium and cyclospora. However, the oocyst of toxoplasma cannot be found in human feces. So these feces, an important point to remember, are acid, sorry, these not these feces, these oocysts are acid fast. These oocysts are acid fast and they are stained by modified Zn stain or Kinneon method of staining. Okay, so they can be stained by modified Zn staining and they are acid fast oocyst. Okay, so I hope I was able to explain the various morphological forms in the previous 3 lectures, including this lecture. And so these are all the morphological forms which can be seen in protozoan parasites. I'll make a small brief quick recap of this video in a small video, so that whenever you want to recap the morphological forms which can be seen in protozoan parasite, you can quickly recap it if you have gone through this video. Okay, so but before going into the recap, I request you to please go through this video, because once you will have a concept and once you will go through it slowly, then you will be able to recall the things very easily during the examination. Don't try to study in hurry. Give the time which is actually needed or which actually the topic deserve, and then try to recall it more frequently by using like videos which I'll make, or by making your own notes, so that during the last minute you don't have to go through the videos or go through the notes which I will give along with the videos. So go through these videos, make a concept and then try to remember it. If anything, if you want to ask me, you can comment me in this video. I'll always answer all the comments. I'll be happy if I'll get the comments in the video. So please comment and ask and let me know your opinion about the videos and the topic, and also let me know that what other topic you want me to make the lectures, if you are liking the videos and the lectures. So thank you so much. All the best. Take care. Bye bye.